Welcome back to First Principles Daily, Episode 10. Starting the week by asking what things should actually cost. Traditional vaccines grow and inactivate whole pathogens, one disease at a time. But messenger RNA hands the cell the genetic instructions and lets it make the antigen instead. For decades, a new vaccine meant choosing a pathogen, growing it in eggs or cell cultures, purifying and inactivating it, then testing the resulting material batch by batch. The entire chain had to be rebuilt for each target. Messenger RNA changed the physical starting point. Once the sequence that codes for an antigen is known, the molecule can be synthesized chemically and packaged for delivery. The patient's own cells then become the production site. That single shift removed most of the bespoke biological manufacturing steps that had set the pace for a century. Vaccine makers inherited a process that began with Louis Pasteur and was refined through the 20th century. The default was to obtain the actual organism or a close relative, propagate it at scale, and then modify it so it could no longer cause disease while still provoking immunity. For influenza, this meant millions of fertilized eggs each season. For other viruses, it meant large bioreactors running mammalian or insect cells. Every new pathogen required its own validated growth conditions, purification protocols, and inactivation chemistry. Regulatory agencies approved each product as a distinct biological entity, so the data package and the manufacturing license traveled together. The result was a finished dose whose cost reflected months or years of living organism culture, extensive quality testing, and dedicated facilities that could not be switched to another product without revalidation. Because the organism itself was the feedstock, any change in the target disease forced the entire upstream process to be re-engineered. That constraint felt inevitable inside the industry. It was simply how vaccines had always been made. The messenger RNA route begins with the observation that the only information a cell needs to produce a protein is the nucleotide sequence that encodes it. If that sequence can be delivered intact, the cell's own ribosomes and endoplasmic reticulum become the antigen factory. The raw material floor is therefore the cost of the 4 nucleotide triphosphates, the lipid components of the delivery vehicle, and the energy required to assemble and purify them. A rough magic wand estimate for a typical 1,000 to 2,000 nucleotide messenger RNA molecule, plus the ionizable lipids, cholesterol, and PEG lipid that form a lipid nanoparticle, puts the commodity input value in the low tens of dollars per gram at current bulk prices. A single vaccine dose contains micrograms of material. The traditional process, by contrast, carries an idiot index measured in the hundreds when one compares the final filled and finished dose price to the cost of the original pathogen seed and culture media. Consider what that ratio actually means in practice. If the seed culture and media cost on the order of a few dollars per dose equivalent, yet the finished vaccine reaches hundreds of dollars in effective production and testing overhead, the multiplier lands in the hundreds. That gap signals that the dominant expense sits in the handling of living organisms rather than in the atoms themselves. The redesign attacked 3 linked steps. First, the sequence itself is now written in software and ordered from a synthesizer. No living pathogen is ever handled. Second, the lipid nanoparticle formulation is a standardized manufacturing line whose inputs are the same regardless of the sequence inside. Third, the quality release tests focus on sequence identity, capping efficiency and particle characteristics, rather than on the variable behavior of a whole inactivated organism. Each of these moves removes a category of cost that's scaled with the biology of the target, rather than with the chemistry of the platform. The key engineering trade-off was proving that chemically synthesized messenger RNA could be translated at useful levels inside human cells without triggering destructive innate immune responses. That required both modified nucleosides and a reliable lipid carrier, both of which had been under development for more than a decade before the pandemic. Once those 2 elements were in place, changing the antigen became a matter of changing the ordered sequence and confirming the new molecule. It did not require rebuilding a production organism and its associated facility. The most visible outcome was the compression of the interval from antigen sequence publication to first-in-human dosing to roughly 2 months for the original SARS-CoV-2 messenger RNA candidates. Manufacturing could be performed in facilities designed for nucleic acid chemistry rather than high-containment live virus suites. The same production line could be redirected to a new sequence with limited revalidation. The idiot index of the final product has dropped because the dominant costs are now the lipid nanoparticle components and the validated fill-finish operations rather than organism culture and inactivation. Remaining hard limits include the need for ultra-cold or improved formulations to maintain RNA integrity. They also include the relatively high cost of current lipid raw materials at clinical grade purity. And they include the regulatory requirement that each new sequence still undergo clinical safety and immunogenicity studies, even when the platform chemistry is unchanged. Those constraints are real. They are not eliminated by the shift to cellular synthesis, but they are narrower than the set of constraints that applied when every vaccine required its own living production system. A product whose cost is driven by the need to grow and handle a unique organism is announcing that the design problem has not yet been reduced to its information and chemistry fundamentals. Once the requirement is restated as deliver the correct sequence to the right cells, the manufacturing problem changes from bespoke biology to standardized chemistry. The same move applies wherever a production step still depends on handling the final physical object rather than its description. Question the requirement before optimizing the existing process. Which other biological products are still being manufactured by growing the thing itself instead of instructing cells to make it? That's a wrap on today's first principles breakdown. The ideas travel further when you pass them on. Send this to a fellow builder. I'm Patrick in Vancouver. See you tomorrow. And if you'd rather watch than listen, find us on YouTube at Nerra Network. Links in the show notes. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own. I'm about to prove